Hello and welcome to my virtual biology presentation. My name is Zana Rizvi and I'm a junior at Rutgers Camden majoring in biology. I'm going to be talking about the research I've been doing called Characterizing Genetic Elements on Conidiation in Neurospora crassa. First, I want to explain what conidiation means. So, conidiation is a multi-gene trait and it's responsible for the production of asexual spores by fungi. Conidiation is a major means of disease spread and is involved with spreading within the host. So, understanding how it's produced is important. It is known that many genes are responsible for conidia production, but the mechanism behind the quantitative production of conidia is not well understood. To identify potential genes that may be responsible for conidia production, first, a QTL analysis was performed. A QTL analysis is used to identify specific regions on chromosomes that correspond with conidia number. It was performed on the N6 population that was generated by originally crossing two parent strains, FGSC 4825 and FGSC 2223. Three major QTLs and several minor QTLs that are responsible for the conidia trait were identified by doing this QTL analysis. Chromosome 1 contains a major QTL, and it was originally used because it had a good potential candidate gene. After identifying the QTL region, it was narrowed down by performing two back crosses. Backcrossing involves taking a progeny strain from the original parent cross with the 4825 genotype and phenotype, which is high conidia production, and crossing it again with the 2223 strain. One of the progeny from this backcross was backcrossed with the 2223 strain again to try to further narrow the region. The region was narrowed down to around 300,000 base pairs on chromosome 1. Figuring out the exact regions responsible for conidia production is an important part of this project because then a list of candidate genes can be found within the region. So using the fungi database, I found 4 potential candidate genes for chromosome 1, and I also decided to look at chromosome 4 and found 1 potential candidate gene. Several knockout strains were found for genes present within the QTL region, and then a conidia counting assay was performed on the strains to see which ones were the most different compared to the wild type. The first experiment showed that strain 11345 had a significant variation from the wild type, which is why it was used in previous research. The second experiment that I performed is preliminary data and has to be repeated. In the future, a transgenic strain test will be performed to determine the sufficiency of the potential candidate gene that I end up using. Sufficiency involves putting the gene back into the organism to help determine if the gene has a direct effect on conidia production. Again, my name is Zanab, and I want to thank Dr. Lee and all the members of the Lee Lab for their support, and I want to thank you for your attention.